Uh, Hi all, hope you can hear me well. My name is Bhavna and I'm pediatric oncologist and staff specialist at the Children's Hospital at Westmead. And welcome to our Advanced Therapeutics Webinar series, where we focus on rapidly evolving space that is advanced therapeutics as it presents innovative and potentially transformational treatments for children now and in the future years. Now, before we get started, I'd like to begin by acknowledging the traditional owners of the various lands on which we are meeting today. Sydney Children's Hospital Network acknowledges that our facilities are built on the lands of Bidjigal, Cambergal, Puramatagal, Cabrogal, Wanagal, and Wadai Wadai. Our services extend across the lands now known as New South Wales. Aboriginal people believe these lands hold the ancestral spirits of those who have come and gone before them. We acknowledge this is, was, and always will be Aboriginal land. Now, today we'll be hearing from Professor David Ziegler and Dr. Neevika Manoharan. Professor David Ziegler is a senior pediatric oncologist, and Dr. Neevika Manoharan is also a pediatric oncologist, both specializing in neuro-oncology and both based at Kids Cancer Center at Sydney Children's Hospital at Radway. Their presentation will explore the future of cancer care for children and young adults. Now, following this presentation, we will move to Q&A session to foster discussion. So please submit your questions throughout the webinar in the Q&A tab on the Zoom controls. And if you see a question submitted that you really want the panelists to answer, please upvote it by clicking the thumbs up sign beside it. I will now invite David to begin his presentation.
Thank you, Bhavna, and I'll just share my presentation. Hopefully this works. All right, so thank you very much for the opportunity to speak and share some of our results. I'm going to speak about our precision medicine program in childhood cancer and end with our latest clinical trial that's attached to that. And then Neve will follow with our CAR T-cell program. So yeah, let's begin with what is happening with our precision medicine. So we really started this now 9 years ago when we started really thinking about precision medicine as in, using all this genomic technology that was becoming available and whole genome sequencing, and how can we use that to find new treatments and new personalized treatments for our children with the highest risk cancers where they don't necessarily respond to chemotherapy or radiation therapy or fail those therapies. And at the time, this was becoming more sort of the trend. Adult studies were being done showing it's possible, but implementing it is difficult. And there were some studies showing, yes, it might be possible in children's cancer, but there's not as many mutations in children's cancer as there are in adult cancers. And it's not clear how many are targetable. There's not as many drugs approved for children with cancer. And so we really, thought we need to do this. How can we do this to make it as successful as possible? So what we thought and what we aimed to do was really try and make this program as comprehensive as possible. So rather than just using a panel of genes, and a lot of the early studies were using a small panel where you test 100 or 150 genes. We thought we'll do that, but we'll also try and sequence the whole genome at the same time and see what we can learn from that. We'll do RNA-seq, so look at gene expression. We'll add it in DNA methylation profiling, which is a more recent technique for helping with the diagnosis of different tumor types. We'll try and grow the cells in the lab and actually see if we can screen drugs, because if we can't find a genetic change, maybe we can find a drug that's active just in the test tube. And we'll also try and develop mouse models for each child and basically try and take all these results, analyse them, bring them to a multidisciplinary tumour board and see if we can come up with a recommendation for the clinician with a new potential treatment for that child. And we thought, well, if even in 10% of patients we can find something that's useful, then this was our sort of bar that we set, that this would be a worthwhile program where we may, for some patients with these highest risk cancers, find a new potential therapy. And, you know, look, I was skeptical. We had a lot of skeptics around the country. This was you know, a new way of doing things, and a really important part of the program was actually surveying both clinicians, scientists, and parents and children as well to understand how we were doing and was this acceptable what we were doing. And, you know, an example from some of the clinicians that you can see here is skepticism, not anticipating a whole lot of miracle wins, to be honest, and concern that we might find a therapeutic target, and yes, there's a drug available, but you can't get the drug, you build hope and you take it away. So we might actually be causing harm with this program. But we rolled it out. You know, the first genome took us about a year to sequence for the first patient. But the team built and the skills built, and we were rapidly able to improve on that. And this was the paper we published now 5 years ago, showing the results for the first cohort of patients that we enrolled around the country. And the first thing we found was that, yes, in 90% of patients, we're actually able to identify the genetic driver of their tumour or drivers of their tumour. And so that was an important finding. And in many of those cases, or at least in some of those cases, there were important druggable drivers. And an example of that was this patient, one of the first patients on the program, who had this very large mediastinal tumour, which was, as an infant, she'd had chemotherapy, it didn't respond, ended up in intensive care with this tumour that was growing, pushing her mediastinum, collapsing her lung. She was on a ventilator, but was becoming increasingly difficult to ventilate and was pretty close to death when we discovered this NTRK fusion. So a novel fusion for which there had just been developed a drug that specifically targets this NTRK gene. No one in Australia had been treated with it. We managed to get the drug company to agree to fly the drug in as quickly as they can. It took a couple of weeks. And she started on this drug, which was just a liquid formulation. She was taking once a day, just like really with minimal toxicity. And the tumour started to shrink within a few weeks. She came off the ventilator. Within 8 weeks, it had much better. And then within 12 months, it was almost completely gone. So this was really the dream that we were looking for, you know, a highly targetable new drug, untreatable cancer, and we're seeing this amazing result. But, you know, this is the dream, but it is not what we see with all patients. And so what we did show, you know, the question is, are the patients benefiting apart from this, you know, occasional miracle? And what we published as well in that study was that, yes, for those patients who we can identify a treatment pathway, and about 70% of the time we could identify a potential new treatment. For those patients who then went on to receive the targeted therapy, actually the majority had some clinical benefit, either stabilising their tumour, about 60 to 70% had stable tumour, and actually about 30% we saw an objective response, so tumours actually shrinking, or in those rare cases like you saw, actually disappearing. So for many patients we were actually able to see a benefit. Importantly, as I said, we were surveying parents and we found that, yes, they were very hopeful, but they weren't really worried. And this, importantly, was even after they received their results, and even if they didn't find something that would help their child or couldn't access the drug, in general, there was really no distress we were causing for parents in this setting. They appreciated both the ability to be able to leave no stone unturned, but also even to be able to help other patients in the future. And similarly, we saw with clinicians an increasing understanding of how the program works and an ability to you know, build it into their clinical practice. And this is some of the change in sentiment, I think, from clinicians around the country. I think that it's really good for us to be able to offer this type of testing here and not have to say to families, oh, we can't do this here. It's pretty world leading. And then even when we talk about the hope that I alluded to earlier, you want to give the family hope, but you need to be careful not to give them too much hope, because that's not actually the endpoint of the study. So clinicians really learning how to explain the study and what the potential benefit or non-benefit may be for their patients. But still, you know, even though we saw we were able to do this, get the results back, get patients onto treatment, there were still many unanswered questions. So yes, we're seeing tumours shrinking, but we know most of these high-risk patients are still ultimately not surviving in the long term. So are we really making a difference there? Are we impacting survival at all? Which patients we are actually most likely to benefit? When you find different potential treatments, which one do you choose? Which one's most likely to help? And when do you start treatment? Do you keep it in your back pocket, or do you start it, as soon as you get these results? And so more recently, we published one of the first really long-term follow-up of these patients on a precision medicine program in paediatric cancer. And really, for the first time, we're able to show that, yes, we are actually impacting the survival of these patients. So when we looked at the first over 300 patients on the trial, and we compared those who got a PGT, being a precision-guided therapy, so a treatment that we recommended based on the genetic changes in their tumour, there was a significant improvement in event-free survival, progression-free survival. So really more than a doubling in that survival. And particularly when you looked at trying to, look at matching like for like, compare patients who got a new therapy, a precision-guided therapy, versus other patients who also got a new therapy, but in this case, it wasn't precision-guided. So it wasn't guided by the genetic changes in their tumour. It was not matched to a gene. It was just a new drug that may be available on a clinical trial or otherwise that their clinician was able to access. And that's where we see a really stark difference, that if you can match the drug to the gene, the survival is almost 5 times better for those patients. So, really showing that there is how we are benefiting patients, not just seeing tumours shrinking, but actually seeing them surviving more in the long term. We were also able to start to answer some of those questions about, well, which drugs do we choose, and look at some of the different genetic changes that we see in the tumour and see that actually, depending what you're targeting, you have different impacts on survival. And so, for example, a fusion, like the patient I showed you with the mass in the chest where we found a fusion, these are really the drivers that that have the biggest impact on patient outcomes, with really a very dramatic improvement in survival if you can target a fusion compared to other mutations or other genomic drivers. And, we looked at a number of prognostic factors, such as the fusion, such as the type of treatment, and putting them together, again, you can see a really significant impact on survival. If you find the right drug, you give it early, you're targeting the right driver, and these patients can do really well. So I think this is really important information that can help clinicians decide which patients to treat and how soon should they treat them. The other group of patients that we looked at, and as I said, we started with these high-risk patients with the worst tumours who really need new therapies. We started getting lots of inquiries from patients, from clinicians, I should say, and even from pathologists saying, well, we've got all these other patients who we'd really like to do this testing on. And this is what we call the rare cancer cohort. So it might be a patient where the pathologist can't make the diagnosis, or it might be one where they can make the diagnosis, but it's so rare that they don't know if it's a high-risk tumour or not, because no one's ever treated it before and no one knows what the right treatment is. Or it might be that they've got a specific gene that they're looking for that there's no clinical test for. So we're actually now looking at this and haven't yet published this data, but have a manuscript in preparation. And the result is, when we started this, we really didn't know, would this genetic testing help these patients? We thought, even if we can help, a tiny number, we opened a cohort, we said, we'll enroll 15 patients, and if we can help 1 or 2 of them, then we'll keep going. And actually, we now analysed more than 100 of these patients and found, actually, for the vast majority, we're actually able to make major clinical impact. And this is the example of the patients who actually we were able to change their diagnosis based on their genomic profiling. In some cases, the pathologist didn't know what the tumour was at all, and the genetic testing was able to make that clear diagnosis. Or in other cases, they thought they might know what it was, or they had a working diagnosis, and that diagnosis changed on the basis of the genetic findings. So really clearly important clinical impact. And in some cases, and actually a very high number of these patients, we were actually to also make treatment recommendations. And this is an example of an 11-day-old baby, a very rare fungating tumour. I'll show you a trigger warning. I'll show you some pictures in the next slide. It's not pretty, but it was very difficult to diagnose. International input was sought. No one around the world really knew what it was. And one of the pathologists overseas said, oh, someone's going to recommend that you do whole genome sequencing on this case, but that's not going to help. I don't think that's going to help. But in fact, it did help. It helped make the diagnosis. And it also, we found, so that this very rare keratin-positive giant cell-rich tumour with this rare fusion. And based on that, we made a treatment recommendation that we thought the drug imatinib may actually help based on that genetic change. And indeed, it did. And here you can see, as I said, this very ugly fungating tumour, single agent oral therapy, had no idea if it would help, but thought it was worth a try. And you can see a very dramatic and almost, again, miraculous response in this trial. So the next question was, all right, we're helping these high-risk patients with high-risk tumours. We're helping these patients with these very rare tumours. Can we help all these other patients who we treat who actually have a good prognosis? And can this genetic testing help with those patients? And really, can we change the way we diagnose patients at the beginning from, what we've done for a few hundred years of looking down a microscope and actually use this genetic testing to help the pathologists, for all children with cancer? And so with our current trial 02, we've gone from just looking at those very high-risk patients to now being available to every single child with cancer from diagnosis in all of Australia. And to do this, we've basically gone from 150 patients a year to now 1,000 patients a year. And how can we do this, in real time and get results back to clinicians without everything falling over and overwhelming the workforce? And the bioinformatics team, led by Mark Cowley and Marie Wong at the Institute, have done an amazing job really semi-automating this with a computational system, essentially, that can turn the work around to analysing this whole genome, all this genomic data, which, as I said, took a year for our first patient. Now, together with the assistance of all this bioinformatics programming, within 48 hours, can help with that analysis. And that basically comes out into what we call a semi-automated report. Clinicians still involved, helping and curations, and there's still a human oversight, but it's semi-automated, meaning that we can very rapidly get these results back out to clinicians for every child with cancer. And we're now looking at saying, are we actually helping pathologists? Are we helping clinicians by giving them all this extra information? And we don't have the answer yet. The preliminary data suggests that we are, but I'll have to come back to you with more. And then finally, the question is, for those high-risk patients, how do we get more of them access to their drugs? And how do we, yes, we're improving survival, but you saw from those curves still the majority, ultimately are not surviving in the long term. So how can we improve that further? How can we get more access to drugs and get them access to better drugs and even better treatments that work more effectively? And so we've built this companion clinical trial that's called OPTIMISE. So up till now, what we've been doing is just giving the results back to clinicians and saying, go get a drug and, see if you can get it from a pharmaceutical company and see if it helps, to now actually saying, well, we'll, through this trial, provide you with a drug, but also try and improve outcomes further by getting new drugs that previously haven't been tested that show more promise, but also focusing on combination therapies. So not just a single agent, but can we actually test new combinations of targeted therapies to further enhance their activity and enhance the survival of these kids? And these are the first arms that we are opening, or in some cases open, and already enrolling patients, and already expanding this internationally. So we're opening this trial now in Canada as well, and hoping through their similar program to also extend this trial internationally. And these first arms are really about targeting the most common pathways that we find to have genetic drivers, and therefore being able to offer the majority, or as many patients as possible, access to these novel therapies. So in summary, this is what we think and we hope is really the new era in childhood cancer, where we're really seeing, and we've shown for those children with high-risk cancers that using this approach, we can double their survival. we are now defining and hope to publish the role in these rare cancers. And now we've gone national for all children with cancer with these rapid reports, and we're seeing, can we actually use this genetic information to help make the diagnosis, stratify patients onto the right treatments, help understand their prognosis, and help change the way we have diagnosed tumours, and bring it into the 21st century. And then, as I said, we're building into that new clinical trials to test new combination therapies. So a huge team behind this, which I don't have time to go through everyone, but just to indicate this is really a national program with collaborators from all around the country, the main hub of which is here at the Kids Cancer Centre and at the Children's Cancer Institute, and a lot of support over the years from a lot of people who've helped make this reality. And I'll just end off before Neve segues into our work specifically on brain tumours, and because I can't give a talk these days without highlighting that the biggest brain tumour conference in the world for children with brain cancer will be here in Sydney next year, where you can learn about precision medicine and more about the CAR T-cell program that Neve will talk about now. So I'll hand over to Neve, and we'll come back for questions later.
Thank you, David, and thank you, Bhavna. I'm going to try and share my screen and talk to you a little bit about our CAR T-cell program, so just bear with me a minute. Okay, so if everyone can see that, I'm going to start by saying thank you to the CAP webinar team for the opportunity to talk about our CAR T-cell study, which David and I co-chair, and we're pretty excited to say that it's the first pediatric brain tumor CAR T-cell program in Australasia, and we'd like to sort of share a little bit about the lay of the land in terms of pediatric brain cancer and immunotherapy, but also some updates on our study. So some, but not all of you may know that pediatric brain cancer is the leading cause of cancer-related death in children in this country. And so while there's sort of 250 to 300 new diagnoses of brain tumors a year, not all of which are brain cancer, and that by definition still counts as a rare cancer, that, you know, really means that up to 300, not only children, but families, communities, and schools are afflicted with these devastating diagnoses each year. One of the big problems that our field in Australia had up until about 10 years ago was that many felt that we couldn't run our own clinical trials here in this country because maybe we didn't have enough patients, maybe we didn't have the expertise to translate our preclinical findings into clinical application. And so how is it possible that we would be able to do things on the same sort of level as our international collaborators, predominantly in Europe and the US? And so hopefully what I'll show you here is the story of how one such study in our CAR T-cell study is now up and running and recruiting patients. So for those of you who haven't seen this photo before, this is Robin Bush, who is George W. Bush's daughter, who in the 1950s at age 3 was diagnosed with acute lymphoblastic leukemia. Now, ALL, or acute lymphoblastic leukemia, is the most common childhood cancer we see and treat. And at the time, her parents were told it was incurable. They flew to New York City for the best available treatment at the time. And even despite that, she died of leukemia 6 months later. Thankfully for our colleagues who predominantly treat leukemia, the cure rates for ALL have drastically changed in that time. So it's gone from being an incurable disease to having cure rates of now in 2025 well above 85%. Unfortunately, for those of us who treat children with brain cancer, those cure rates and that trajectory has really not been the same. And despite countless amounts of research and clinical trials, we're still really struggling to shift the bar for certain types of pediatric brain cancer. In particular, there's a real dichotomy between what we regard as low-grade tumors, what some may call benign tumors, which are sort of grade 1 tumors, versus those that are considered more high-grade, which are sort of grade 3 and 4 tumors, which, as you can see here, have a really devastating prognosis, and probably none worse than that sort of green line that you can see, which is high-grade glioma. High-grade glioma, segueing into this, which is diffuse intrinsic pontine glioma or diffuse midline glioma, is the most aggressive type of high-grade glioma we see, and unfortunately it makes up about half of children with pediatric high-grade glioma. There's lots of barriers to effectively treating it, but one of them is that it occurs anatomically in the midline of the brain, where there are crucial structures that really are important in respiratory function, cardiac function, your cranial nerves, that mean that these tumors typically cannot be resected. They're defined by an epigenetic alteration in the histone of the gene, so the coiling and uncoiling of DNA. And about 20 to 40 children in Australia are diagnosed with this devastating disease each year, with a median survival that's less than a year. So effectively, they're incurable and have a short survival. Radiation is still the only standard therapy available. And there have now been over 250 clinical trials in this disease, but unfortunately, none of them have yet been curative. And so from that, we get to how do we sort of bring CAR T cells to pediatric brain cancer? Well, the field of pediatric oncology was really revolutionized when patients with CD19 positive leukemia were treated with the first set of CAR T cells. This is the first ever patient to be treated with a CD19 positive CAR T cell for leukemia, who is now a long-term survivor over 10 years later. And it really revolutionized the care of these highest risk leukemia patients. So it made us in solid and brain, solid tumors and brain cancer wonder, how do we translate this work to our patients? But what is a CAR T cell, I guess, for those who aren't as familiar with it? We call it a living drug, but what it really is where you take the patient's T cells and you genetically modify them. So you transduce them with a vector that sort of, in this case, our case is a gamma retroviral vector. It encodes and sort of genomically integrates with the DNA of the T cell. That then means that the T cell can express particular antigens and antibody constructs that mean that it can detect, in this case of CD19 leukemia, a matching antigen. And then it also has a co-stimulatory domain. That means in the ideal world, it binds to the leukemia cell in this case and causes single-step binding and cell kill. Unfortunately, though, in DIPG or high-risk brain cancers, there's not just one easy target that was the clearest path forward. So we put this together for a recent review paper, and these are all the current open pediatric brain cancer CAR T cell studies. And there are two main areas of focus that people are interested in terms of the target, and one of them is GD2. So our study is that bottom one there, and there are a couple of our colleagues and collaborators around the world who are also studying GD2 CAR T cells for pediatric brain cancer. So why GD2? So GD2, or disialogganglioside, is a glycosphingolipid, and basically it is expressed at low levels in lots of healthy nerve cells, but also expressed in many childhood cancers. This cell surface screen done out of Michel Monge's lab was the first to show that DIPGs with the histone alteration uniformly expressed high levels of GD2, and those that didn't have the histone mutation basically had no GD2 expressed. So that, combined with Crystal Makel having developed a GD2 CAR, resulted in this really nice mice work that showed that giving GD2 CAR T cells to DIPG-bearing mice resulted in, by day 14, most of the tumors were gone, and they were able to sustain that response for an unexpectedly long time. However, what people don't always recognize is that only the mice that survived their GD2 CAR actually had this remarkable response, and many of the mice succumbed to the toxicity of the CAR itself. So that brought about real concern about how are we actually going to be able to give this sort of therapy to adults, but more importantly, how can we give it to children? Which brings us to our study, Levi's Catch, which is leveraging chimeric antigen receptor expressing T cells for children with diffuse midline glioma, which I co-chair with David Ziegler, and it is our phase 1 study. So this study is quite beautifully named after Levi Wheeler, who is a heroic young man who was treated at our institution and died of DIPG. And his amazing family have been really instrumental in being able to fundraise and with their community to be able to allow us to now open and run this clinical trial. To sort of just say one more thing on GD2 CAR T cells, I guess, is that this sort of therapy is of interest in lots of different cancer types at the moment. So we're looking at it in diffuse midline glioma, but others are looking at it around the world for childhood cancers such as neuroblastoma, osteosarcoma, and in adult cancers in glioblastoma and melanoma. But if you look at the GD2 CAR T cell technology column, you'll see that not all of these CAR T cells are made equal. They're not the same. So ours, pleasingly for us, is a third generation CAR, so a slightly more advanced CAR, and also has slightly different co-stimulatory domains to some of our collaborators around the world. So as I mentioned, we have a third generation CAR with a vector that was derived from Baylor College of Medicine. So this is truly both a national and international collaboration. It has 3 co-stimulatory domains, CD3 zeta, CD28, and OX40. And one of the most exciting things about it in my mind is that it has a inducible caspase-9 suicide switch, which means that in the event that a patient experiences severe toxicity, we are able to administer a drug that actually switches the CAR off, which is pretty exciting. The other probably big positive thing for our study is the manufacturing cost. So our collaborators in the US quoted me that their CAR T cells cost about $50,000 to $100,000 to manufacture per CAR per patient. And so we're able to do that just under 10,000 Australian dollars per patient at the moment. One of the big things to really mention is that it is a true labor of love being able to get a CAR T cell study from conception to opening and recruiting, because it is a immunotherapy-based trial and it's a cellular therapy-based trial. So there is a whole number of hurdles to jump. And this is also to say thank you to the entire team who make that happen, both at Sydney Children's Hospital, but importantly, our collaborators in Adelaide who manufacture the CARs, Michael Brown and Tessa Garger. So Levi's Catch is a phase 1 study. So by definition, our goal is to find whether it's actually safe and is it possible to manufacture these CARs for patients with DIPG, and is it safe to give it? But what everyone wants to know is, can we do something in terms of, are we able to generate tumor responses in these patients, and can we sustain them? Our study differs a little bit from some other GD2 CAR T cell studies in that we give, together with the CAR, we give a drug called bevacizumab, which is an anti-VEGF antibody. Our goal with that is to actually reduce some of the swelling in the brain and around the tumor that can happen with CAR T cells, but also with the theoretical hope that it might improve penetrance of the CAR T cell into the tumor itself. What does it mean at a patient level? Well, at a patient level, patients come to us at Sydney Children's Hospital for what is called leukapheresis or T cell harvest. Those T cells are collected, the remainder of their blood goes back into their body, and the T cells are sent to Adelaide, where CAR T cells are manufactured by Tessa and the team. They are ex vivo expanded, cryopreserved and sent back to us in Sydney. We give the patients lymphodepleting chemotherapy to really suppress the immune system enough that the CAR T cells are allowed the opportunity to propagate and migrate across the blood-brain barrier. And then we administer the CAR either IV or intravenously, or most recently, we also have approval to administer it directly into the CNS by direct injection into a reservoir in the head called intracerebroventricular administration. And then we watch the patients very closely. So we are pleased to say that we've now recruited 12 patients on Levi's Catch, and we are actually currently infused, well, we've just infused this week our 6th patient on study. One of the most positive things so far is that we've been able to manufacture successfully a CAR for every single patient on the study. Unfortunately, not all of the patients have gone on to receive their CAR. The first iteration of our study had to be very careful about for whom we were giving these therapies to due to the risk of toxicity. So if patients had a disease progression or became too unwell, they were unable to go on and receive their CAR. But as we've learnt more about sort of selecting the right patients for this sort of a study, we've been able to infuse more and more of the patients enrolled. We've also completed out our first dose level 1, and we are now on our sort of interim next dose level, and that's where our current patients are being infused. One of the biggest concerns about CAR T-cell studies, but particularly GD2 CAR T-cell studies, because GD2 is expressed on nerve cells, is toxicity. And that toxicity can really be divided into 2 main groups. One is systemic toxicity to your whole body, and the second is toxicity to your brain or your brain around the tumor. So cytokine release syndrome is systemic toxicity that sort of mimics sepsis. You get fever, hypotension, hypoxia, and it's mediated by the interleukins that are released in the processes of the CAR T cells expanding and enacting their function. But the one that's probably most specific to us with GD2 CAR T cells in the brain is this phenomena called tumor inflammation-associated neurotoxicity, or TIAN, which is slightly different to the neurotoxicity seen in leukemia CAR T cells called ICANS. The particular concern for us is that the CAR T cells can generate swelling around the tumor itself. And as I mentioned, DIPGs or DMGs are tumors that occur in the midline. So swelling in these really important parts of the brain have the risk of becoming an emergency, and that you might be in a position where the patient develops hydrocephalus or the need to manage raised intracranial pressure. Our colleagues in Stanford who collaborate with us on this study at the end of last year published their first sort of findings out of their GD2 phase 1 CAR T-cell study, and probably 2 main things came out of this publication. The first was some really exciting tumor responses, patients responding to the products, and also having some sustained responses out to sort of 35 months, which for this disease is definitely outside the historical controls. But the second main thing was that they did see toxicity, both systemic and neurotoxicity. Almost all of their patients had CRS and TIAN, and some of the patients who had tumor inflammation-associated neurotoxicity went on to be quite unwell in the intensive care unit, requiring ventilatory and cardiac support. We've been pleased to say that we've completed out our first dose level, and we actually have no serious dose-limiting toxicities associated with the CAR T cells. We had 1 patient who sadly progressed on the study and had some toxicities related to that, and some expected cytopenias related to the fact that we give lymphodepleting chemotherapy. But other than that, we've actually, based on these findings, have been able to modify our protocol to allow patients to be able to be discharged earlier. And for those who have patients who are coming to us from around the country, it means that they may actually be able to return home sooner as well. One of the big questions with CAR T-cell studies is, can you get the CAR T-cells into where you need them, which is in our case, the brain, and can you keep them there? So we've been pleased to see that despite administering the first dose IV for all of our patients, both in the blood and the CSF, we're seeing high levels of the CAR transgene at day 14. By day 28 and by day 42, we are seeing a drop off in that. And so one of our goals is to try and improve the persistence of these CAR T cells in the brain. The way we've overcome that currently is by sequential administration, which means that each patient is allowed to get a second, third, fourth CAR T cell after 6 weeks from the previous one, if they want to, and 2, if they're responding and tolerating it. And excitingly, we've actually also infused our first patient intracerebroventricularly, which means directly into the brain through a reservoir, and we've done that without any major toxicity. One of the things that we're really looking at is to see whether there is any differences in the patients that respond versus those who don't, because one of our key goals is to try and offer this therapy to those who are most likely to respond whilst still making it accessible to everyone for whom it's safe. And so what we are seeing is in the 2 patients where we've had the most impressive radiological responses, that they do also have the highest levels of CXCL10, IL-1 beta, and CCL2, so pro-inflammatory cytokines. This is in the blood, and that's also replicated in the CSF. Our first patient who progressed on study had a completely bland cytokine profile. Now, these are the 2 patients for whom we've analysed radiological responses who have had quite remarkable radiological responses, but I'll preface this by saying they are also patients that have received radiotherapy, and we do also give bevacizumab, but we're very excited to see some early radiological responses that look quite impressive and also look to be sustained for longer than what we might expect from traditional radiotherapy responses alone. So before I finish, I think it's clear that in a disease where there's been 250 clinical trials and none of them have yet cured this disease, that no one single strategy is likely to be curative on its own. So I think our goals are looking future forward, that we want to see what we can combine CAR T cells with in the future, how we can actually maximize the function of our CAR T cells. So our next study is looking at integrating IL-15 into the CARs, but really, how can we best combine these therapies for patients in the future? And do we need to do things like offer lymphodepleting chemotherapy, or is that not the right strategy? And this is very much something that we're looking at moving forward. But for me, the major impact of this study has been that CAR T cell studies for brain cancer were not at all available in Australia or in Australasia, and patients who wanted to access it through the US the US were not taking international patients. So patients like this family on the study who wanted to share their story have really said that this study has been able to offer hope to Australian children, families, and entire communities with an incurable brain cancer, that they're now able to access cutting-edge therapy close to home. So to finish, Levi's Catch is the first paediatric CAR T cell study in Australasia. We've manufactured products for all 12 of our patients to date. We've given it IV and directly into the brain without any toxicities at the first dose level. We think we've seen a differential cytokine profile between those who might respond and those who might not, but that's early days. There's issues to overcome with persistence, but we really are excited to be able to offer this therapy to Australian children with this incurable brain cancer. I'd like to thank the whole Levi's Catch team led by David and our colleagues Michael Brown and Tessa Gargett, as well as our amazing trials team, but most importantly to Levi's Project, and really to every patient and family who is enrolled on what is a really cutting edge but potentially scary trial, and it speaks to the heroism of these amazing families with this disease. Thank you. I will stop sharing.
Thanks, David and Neve, for that both great presentations. I'll introduce Professor Geraldine O'Neill, who's head of our Children's Cancer Research Unit at Westmead, who will facilitate the Q&A session today.
Thanks, Bhavna, and please do put your questions into the chat so that I can present them to the presenters today. So thanks both for really fantastic and I have to say very clear presentations. Very much appreciated. So if I might start off the questions with David. So firstly, I just wanted to say how great it was to see that OPTIMISE trial is opening. That's going to be really important, I think. But I wanted to ask possibly a slightly provocative question, I guess. When you see something like this kind of diagnostics, there's an improvement in the diagnostics using the technology that you're using. Do you think this means that there's going to be a time at which we can sort of drop off some other diagnostics? And, because you just think, might this help sort of fund this kind of activity going forward?
Yeah, so, I'm always wary about saying we'll replace pathologists because, and I don't think we will. I think this will really supplement what they do. And very commonly now we get pathologists saying, here's what we found, we need to wait for the molecular before we integrate it into our final diagnosis. So they're already using that. The WHO classification for brain tumours now has so many genetic components to it that you really need the genetics, just to fit into the WHO classification. I think what I think is that yes, there's the potential for the sort of the whole genome and this very broad and in-depth testing to replace looking for single genes or looking for for example with neuroblastoma where the chromosomal changes are so important prognostically. You don't have to do cytogenetics because that you can see it on the whole genome sequencing. So yeah, I think you can potentially replace other genetic tests, and the cytogenetics is probably the most low resolution genetic test that we have. Just looking at the chromosomes, and the beauty of this is that it's all there. So even if you don't have to keep developing new tests because someone's discovered a new gene, you can just basically add it into your virtual panel and say, oh, well, now this gene's important. Let's make sure we're pulling that out of the curation.
Great, thanks, David. There's a couple of questions that have come in the chat for Neve. And Neve, if I could just start by saying, Levi's catch, it's so exciting, and I think we're all cheering for that to do as well as possible. So thank you for talking about it today. So the first question comes from Steve Alexander from Children's Hospital. He says, did you have any patients where you weren't able to manufacture the CARs to a level that would be required for administration?
Yeah, that's an excellent question. Thank you for asking it. We've been really fortunate and also lucky with our collaborators in Adelaide that we've had no patients for whom we haven't manufactured CARs yet. In fact, for every patient on the study, we've manufactured an extra CARs. So if they are able to receive subsequent infusions, we've actually got CARs banked for them to receive. The other thing at a patient level is we've been able to collect enough T cells in one single day and sitting for each patient. So that means they don't have to sit on the apheresis machine for multiple days to collect enough to make the CARs. So at a patient level as well, I think what we've seen so far is that the families on the study have found it doable, even for those who have come to us from different parts of the country.
Fantastic. And Michael Schum, who's from the Advanced Therapeutics program, he's asked, and I'm interested in this question very much as well, how you've been able to manufacture the CAR T cells at a lower cost compared to overseas centres.
Yeah, that is as always when you put that in the slide deck, it's like asking for that question, isn't it? We're very lucky in Australia because we've got a permissive regulatory environment compared to our colleagues in the US. So we manufacture in an amazing lab that has GMP-like conditions, but not GMP conditions. And I think that needing GMP conditions really adds a huge exponential cost. But the other part of it is that if you're not seeking to have the intellectual property to sort of then become a mini pharma to then make this sort of a whole line of things in the future, you can actually manufacture and deliver this at a lower cost. So what it's meant for us as a study is that we're able to enroll more patients, and also it means at a per car level, we've got more cars banked for each patient to receive subsequent infusions. So yeah, thank you for asking it. We've been pleased with that cost.
And someone has added a comment. Sorry, David, did you want to add something there? It looked like you're ready to jump in. No.
I agree with whatever Neve says.
Okay, great. Someone has also just mentioned that, of course, there's moves to using fluidics to, improve the kind of manufacture process. And of course, I guess we will see the price come down as time progresses anyway with these kind of advances. Robert Bowie has asked whether there's any benefit from neurosurgery from these patients. So again, I think that's for you, Neve, regarding the DRPG patients.
Yeah, I think it's important. We have the reason the study is currently open in Sydney is that, so for DIPG, it used to be traditionally diagnosed without a biopsy, and now the field is moving because of, as you've seen with what David's presented, we're finding more targetable things in all brain tumors or many brain tumors. So we're requesting, or for those who don't have classical radiological appearing DIPGs, that the patients have a biopsy. But at the level of neurosurgery for the study is actually, we collaborate very strongly with our neurosurgeons here, because the raised intracranial pressure that you can develop as a side effect of the CAR T cells, if you don't have a neurosurgical team willing to help you in your time of need if the patient becomes unwell, it's hard to run a higher risk study like this one. We've been lucky that we haven't had any major toxicity yet, but we also have a very engaged neurosurgical department who are ready to help us if that should happen.
Keep the neurosurgeons on side. I like that.
Absolutely.
So David, a question for you. When do you think we will know what impact the Zero program has in kids with lower risk cancers?
So we're in the process of really analysing the first 1,000 patients who've come through on that program. So hopefully any day now I'll be able to give you a definitive answer. I can tell you, well, you've just started by just asking the clinicians, has this been useful for you? And for the majority, the response has been yes. It's helped either, in a smaller percent of cases, change the diagnosis, probably less than 5%, but in many cases it's helped with risk stratification. So we found genetic changes that have meant, oh, this is a higher risk tumour, I need to treat more aggressively with chemotherapy. So it's not, for most of these kids, it's not about, we're going to pull out the magic bullet and stop chemotherapy, but it's about using that information to, confirm suspicions, find that genetic change that helps with the WHO classification, or refine diagnosis and prognosis. And so, that from just asking clinicians, we're seeing that for many of these patients. And interestingly, even, an example is the low-grade brain tumours, where we had some clinicians say, oh, I'm not going to enrol those patients because they're low grade, we can treat them with surgery. And that's an example we're actually finding some of the greatest benefit, because these low-grade tumours often have targetable genetic drivers, and even if you don't use that straight away, it's really useful information for clinicians. I don't have the full numbers, but I think that's coming and is looking hopeful.
So we have an interesting question from Morag White, who says, thanks both of you for fantastic talks. She wants to know from Neve, do you give lymphodepletion with your subsequent infusions? It's a very interesting question.
Yeah, she's asked one of the pivotal questions in the field, I think, because I think depending on who you asked, you might get a slightly different answer. I think the subsequent infusions with IV infusions, lymphodepleting chemotherapy probably has a more clear role. You suppress the immune system, But there's been some debate about when you're administering directly into the brain, an area which some people regard as a sanctuary site for immune cells. Is there any role in giving lymphodepleting chemotherapy? One of the benefits of collaborating with our colleagues at Stanford is they've shown some quite nice early work that maybe there is a role, even when you're giving it directly into the brain, that giving lymphodepleting chemotherapy may in fact help your propagation of your CAR. The counter to that is we can't keep giving lymphodepleting chemotherapy every 6 weeks till the end of time. So we are sort of examining at each patient level as to what is the best way forward. We're certainly giving it with IV administration at the moment. And the jury's really out as to what the best way forward for ICV ongoing is, because as Morag will know well, we can't, there's a tolerance that your bone marrow will at some point exceed with repeat lymphodepleting chemotherapy.
Geraldine, we have got time for maybe one more question.
Okay, so there's no questions in the Q&A, but I have a question, maybe a slightly harder one. I'm just wondering about the addition of bevacizumab in the CAR-T trial, because it looks like you don't have an arm with bevacizumab and one without. So, you know, are you worried that that's going to muddy your conclusions at all?
That is an excellent question. And I think we're going into this very, you know, eyes open that bevacizumab is a agent that can reduce swelling. It can reduce the swelling that's there after radiation. And so when we're presenting our radiological responses, we're very clear to say that it's important that you declare your confounder. But I think two things. One, if it's allowing our patients to receive the CAR and tolerate it better because they don't get as much toxicity, if you can control the swelling, that will be for the betterment of the patients in the study. And two, I think we know that bevacizumab doesn't treat or cure DIPG. So if there, you know, when we're seeing these more sustained radiological responses, I think there is a good signal there that maybe there is something that that is beyond the bevacizumab itself, but certainly it has been a contentious area, and it does set this study a little apart from some of the others.
And I'll just add, we're selecting patients. So if they have a lot of swelling and edema, and these are the patients you'd expect that bevacizumab might improve things. So we exclude those patients because they're the ones we're worried about that you'll get, you know, when you give the CAR T cells, that will get even worse and it'll be too dangerous. So the patients that we're enrolling aren't the ones that you'd normally expect bevacizumab to be doing much in any way. And like Neve said, I think we have to just think of it as a whole package. And, you know, if we're seeing, you know, this is an incurable disease where nothing works, if we're seeing that this package with the CAR Ts, the lymphodepletion, the bevacizumab is non-toxic, and we're seeing tumor responses, and that's, you know, that's what matters in the end.
I think we're running out of time. So thanks everyone for excellent questions and joining us today. Now the video of this will be available on SCHN's YouTube channel, and there'll be a short survey when this webinar ends. If you could complete that, that'll be great. Thanks again, David and Neve, for a fantastic webinar and great sharing, Geraldine, Q&A session, and Kids Advanced Therapeutic Program contributions, including Luminous Alliance, and most importantly, the audience who participated well. And then stay updated for the next webinar. You'll get it in your email.
Thank you.
Thank you, everyone.